All right, great. That's all set up. So I guess we're now on the record.
Okay, let me post that and then we can get going. Oh, we're still a minute early, actually.
All right, for everyone who's just joined up, welcome. We'll get started in just a minute or so.
Okay, that's all good from this end.
Johanna, how do you feel about, do you want us to ask people to keep their cameras on, or do you have any preferences there?
Whatever people prefer.
I guess you're probably used to it at this point.
Yeah, with the students never having their cameras on, that's for sure.
I mean, I wouldn't, right? Because like I'm watching half the things, you know, in bed or maybe in lab doing other things. And
yeah.
Not that that's the best, but you know, it's the reality.
Okay, I think we can get started. People are late minute joining now. Okay, hi everyone. Welcome to another week with another migration seminar. We're really delighted to have Johanna Ivaska as today's speaker. Johanna is Finnish Cancer Institute K. Albin Johansson Research Professor and a Professor of Molecular Cell Biology at the University of Turku. She's an EMBO member, editor of JCB, and on the editorial and advisory boards of various other journals. She started a research group in 2003, and her group is interested in integrin-mediated cell adhesion and migration, cell matrix interactions, and mechanosensing. And today she's going to be talking about forces and integrin activity during cancer cell migration. So take it away, Johanna, thanks.
Okay, thanks very much, Adam and Jennifer, for the invitation. It's great to get a chance to talk and also see at least very familiar names here in the chat or the Zoom. Okay, so I'll share my screen. Okay, is this all right?
Looks great.
Okay, so as Adam mentioned, I'm going to be talking about regulation of integrin activity and also forces and how they impinge on cancer cell migration. And so as the name of my lab sort of tells you, cell adhesion in cancer is what we're interested in. Oops. And so integrins are probably very familiar to most of you. They are these abundant cell surface adhesion receptors that bind to extracellular matrix ligands outside the cell. And then inside the cell, their short cytoplasmic tails engage actin and numerous proteins in the integrin adhesion complexes. And they form these turning over dynamic structures in a fibroblast that you see here color-coded from a time-lapse video called focal adhesions. Integrins obviously regulate a number of important things, but for the scope of this talk, the main important things will be migration, invasion, and metastasis. So in a regular sort of fibroblast-looking cell, I suppose you can find three categories of adhesion complexes at least. So in the leading edge, in the lamellipodia, you have these transient, short-lived small adhesion structures called nascent adhesions. In these structures, integrins are already getting activated by tailin. They can already initiate signaling, but they're not connected strongly to the actin cytoskeleton, and they're not sensing force. Then in a force-dependent manner, these adhesions can mature into focal adhesions, where a number of integrins engage and cluster to bind matrix ligands, but also recruit a number of signaling proteins here, just depicted with this sort of pink ball here. And then especially in fibroblasts, but also endothelial cells and even some cancer cell types, these focal adhesions can then further slide back tailin is replaced by tensin, and these structures mature into fibrillar adhesions. And these adhesions are very important, for instance, for laying down the fibronectin matrix in ECM remodeling. Integrin activity is regulated on the plasma membrane. In some cell types like blood platelets and white blood cells, almost all of the integrins are fully inactive, and then they're all activated at the same time. Whereas in adherent cells, there will be subcellular localizations of active and inactive receptors, I'll describe you in a moment. But in any sort of adherent cell type, beta-1 integrins will be inactive, predominantly on the plasma membrane, and there are some inactivating proteins that I'll get to in a bit that facilitate this conformation. Intracellular signals that then activate tailin cause tailin recruitment to the integrin tail. The integrin is then extended and transitions into a primed active state. And this can then further transit into a fully active ligand-bound extended conformation, where also actin becomes engaged to the tailin, signaling is initiated, and as I mentioned, then in response to force, this matures into a fully clustered integrin-containing integrin-associated integrin adhesion complex or focal adhesion that is strongly coupled to the actin cytoskeleton, and also initiates downstream signaling to activate focal adhesion kinase SARC, PI3 kinase AKT, and the ERK-MAP kinase pathway. Integrin activity regulation is critical for human health, and there are a number of diseases that are linked to impaired or dysregulated integrin activity. So mutations in kindlin, which is an important integrin activating protein, lead to this Kindler syndrome, which manifests, for instance, in keratinocytes. If you have impaired ability to activate integrins from your leukocytes, you can have a condition called leukocyte adhesion deficiency, which manifests in unwarranted inflammatory conditions throughout the body. If your platelet integrins fail to activate, you have bleeding disorders like this one here. And also rheumatoid arthritis and cancer, for instance, are linked to abnormal regulation of integrin activity. What's been known for a long time is that the active integrin is supported by recruitment of tailin, as I explained. But when we started to work on this a decade ago, it wasn't really well understood whether there's anything that maintains integrins inactive or whether this is just a state they transition into when tailin is absent. And since we thought this is an important question to address, we launched some large-scale RNAi screens to try to look for regulators of integrin activity, and some of these have been published. And for the sake of this talk, I thought I'll just mention a few of them to highlight the fact that active and inactive integrins are actually spatially differentially localized in cells, and also the integrin activating and inactivating proteins are differentially localized. So a decade ago, we published this protein called sharpin, which we identified as an inhibitor of integrin activity. And as you can see from this now very dodgy looking imaging actually, is that sharpin localized to these ruffling areas of a cancer cell. And this co-localized strongly with inactive integrin, which is here recognized by a specific epitope recognizing antibody. And these structures were further up and away from the focal adhesions that are recognized here by the active epitope antibody. And so what we showed in this paper is that sharpin actually binds to the integrin alpha tail and indirectly competes with tailin recruitment, and therefore it favors stabilization of this inactive isoform. And the link to cell migration was such that if you remove sharpin and you have more active integrin, you also start to see higher migration on low ligand densities, whereas on high ligand densities, the integrins effectively actually got stuck and the migration was slowed down according to this sort of biphasic curve of integrin activity and migration speed. Another important situation where integrin activity regulation, as I mentioned, is important is in the process of leukocyte homing in response to inflammation. And 2 integrins on the white blood cells, alpha-4 beta-1 integrin and LFA1 or alpha-L beta-2, are particularly important at this sort of step where the leukocytes form firm adhesions and they transmigrate through the endothelium. And in contrast to adherent cells where the inactive integrins localize to the ruffling areas, in white blood cells, there are these dissociated tails called uropods. And lovely work from Nancy Hogg, already quite a long time ago, has established that inactive LFA1 localizes to the tail. And what we were able to show is that in most white blood cells and also humans, sharpin localizes strongly to the uropod. And if we isolate leukocytes from sharpin deficient mice or silence it from humans, we actually start to see these very long retracting tails that are now fully adherent instead of this integrin inactive uropod tail. And so sharpin localizes to inactive integrin areas, which are different localizations in different cells, and then spatially contributes to maintaining integrin inactive in these areas. Another protein we pulled out from the RNAi screens is a protein called Shank. And first we thought that this would probably be false positive because when we looked at all the literature that was published on Shank, it was all only focused on the central nervous system. Shank is highly mutated in human autism, and it's been studied for its role in the postsynaptic density in how it regulates, for instance, actin and dendritic spine morphology there. However, when we looked at this and we screened it in all of these cell lines, and you can see if it's red, we have increased integrin activity when we silence Shank, it was actually a hit in quite many cancer cell lines. Furthermore, it's actually ubiquitously expressed in many different cell types and tissues outside of the central nervous system. And so we thought it would be interesting to study whether it does anything to integrin activity, for instance, in cancer cells. And what we were able to see is that, again, the integrin inhibiting protein strongly localizes to these ruffling areas that were positive also for inactive integrin stained here with MAP13. What we were also able to show is that silencing of Shank impaired or had a strong effect on all these integrin functions. So if we silence Shank, we have increased adhesion to collagen fibronectin mixture. We see increased cell spreading. These are micropattern 9 micrometer lines, and the longer the cell is, the further it has spread. We also see increased random cell migration, and we also see increased invasion in an inverted 3D invasion system. Depending on the cell type, the predominant form is either Shank1 or Shank3. That's why it just says Shank here. So it's Shank1 in 231 cells and Shank3 in HEC293 cells, for example. The mechanism turned out to be that, again, Shank is impairing tailin recruitment. So what you could see in this video, let's try to play it again. Oops. Is that the Shank silence cell on the right-hand side has much faster tailin recruitment to these forming lamellipodia than the control cell on the left-hand side, where the tailin is recruited to these bigger, more slowly turning over adhesions. And so what we were able to show was that, in fact, the N-terminal structure part of the Shank protein is a RAS association domain. So it structurally makes the same fold as RAS associating proteins. And it has the capacity to bind to active RAP1. And active RAP1 is the key step in integrin activation. So RAP1 recruits RIAM and tailin, and these are necessary for integrin activation. And now Shank1 can recruit the active RAP1, bind it with fairly high affinity, much higher than what's been published for RIAM. And in this way, it can stabilize the inactive integrin by sequestering RAP1 away from the activating system. And as a result, it then suppresses adhesion formation, spreading, and migration. So One of the first take-home messages from the talk is that integrin inactivators don't go to focal adhesions, but they go to areas where integrins are inactive, such as ruffles and the uropod in the white blood cells. Now, is this the case for all adhesion types? One of the adhesion types we've been very interested to study are Filopodia adhesions. And in Filopodia tips, you can find these very small adhesions that stabilize the Filopodia tip and are essential for Filopodia-mediated cell invasion and probing of the ECM. And so in this old video that Guillaume already did during his PhD, you can nicely see how the Filopodia drive the navigation of the cell when it's migrating through cell-derived matrix. And one of the key proteins that induces Filopodia is an integrin binding motor protein called myosin 10. So the myosin 10 has a firm domain, like many proteins that bind directly to integrins, and this site can bind it directly to different beta-1 tails. If you overexpress myo10 in a cell, you can see this strong induction of Filopodia. And here from the scanning EM, you can appreciate that in U2OS, for instance, if you overexpress myo10, you're going to get quite prominent Filopodia formation. What we've seen is that Filopodia actually give rise to focal adhesions. So what we see is that when a new Filopodia shoots out, which will happen in a bit, here, you can see that paxillin starts to slowly accumulate at the base. And then the Filopodia actually matures into a focal adhesion. And what we sometimes see is that this is a subsequent process. So you have a Filopodia shooting out, maturing into a focal adhesion, and then the same structure shoots out another Filopodia that again matures into a focal adhesion. And this might be a way to get directional motility in the area where you can stabilize your Filopodia. And one of the mechanisms we've published is that there are calcium channels and tailin recruited to the Filopodia tip. This is necessary for the stabilization of these structures and the subsequent maturation into a focal adhesion. However, what we haven't really known very well is how is integrin activity regulated in Filopodia, apart from the fact that tailin can be recruited to Filopodia tips. And what we also know is that these Filopodia align with focal adhesions. So first of all, Filopodia are almost always connected to a focal adhesion at the base. So this is the Filopodia tip. I'm sorry, it's a little bit dim. And it's connected to a focal adhesion. And if we do super resolution traction force microscopy, the Filopodia tip forces are so low that we can't detect forces in the tip. But what we do see is that the forces in the base in the focal adhesion very nicely usually align with the direction of the Filopodia, and that's just the quantification. So they might be somehow mechanically linked to each other. We've tried different approaches to understand what is the composition of the Filopodia tips. We've tried different mass spec approaches, and Guillaume's laboratory, now that he's an independent PI, is continuing those efforts. But in the meanwhile, when Guillaume was still a postdoc in the lab, he decided to take a hard-working approach at this. So what he did was he took over 80 proteins that have been published to be in the adhesome. He tagged all of these with a GFP, expressed them one by one in cells, and did super resolution structural illumination microscopy to define which ones of these localized to Filopodia tips, to see whether this would help us to understand how those adhesions are regulated. And so this is the data that was published in 2019 in Current Biology. So you can look for your favorite protein. But what he did for each of these proteins was that he drew lines along the Filopodia, and then he mapped the intensity of the protein along the Filopodia shaft. And all of these heat maps are the average intensities of hundreds of Filopodia. And so what you can see is that some proteins localize very, very strongly to the Filopodia tips. Some other proteins, like esrin, for instance, here is predominantly in the shaft, not at the tip at all. And then quite a lot of focal adhesion proteins actually never go into Filopodia. And so what we were able to conclude from this study was that Filopodia adhesions have a unique composition. So some of the players are the same. So for instance, we see tailin at the tip. We see also kindlin recruited to the tip, and ICAP, which is an integrin inactivating protein. However, for instance, focal adhesion kinase and paxillin are not localizing to the Filopodia tips. P130Cas is one of the very first proteins that seems to go to the Filopodia tip right at the time when MAIA-10 goes there. And what we were actually able to show is that P130Cas is an important mechanoregulator of Filopodia in response to stiffness, but I don't have time to go into that. But if you're interested, you can look at that in the paper. So one of the big I guess, generally accepted concepts in the field has been that myosin 10 transports cargo to Filopodia tips. It's a motor protein, it walks along actin, it can bind to integrin, but also other cargo like adherins and actin regulators. And the idea is that because it concentrates to the tip, the idea has been that one of its functions is to transport all this stuff to the tip. However, now that we had the SIM microscope that we could use to really nicely image in detail the composition of Filopodia and localization of proteins along Filopodia, we made a very interesting observation. And this was the fact that integrins are actually not transported to Filopodia. So if you look here, the inactive integrins decorate almost the whole Filopodia, all the plasma membrane and the whole shaft, and already also to the tip. So they don't need to be transported there because they are there all the time. However, what we did obviously see, and this was in line with previous reports, it was that the active integrin mainly accumulates only at the tip. And here again, with the same sort of line profiling approach that I described, you can see that with 2 active integrin reporters, the active integrin preferentially accumulates at the tip, whereas the inactive integrin is found throughout the whole shaft of the Filopodia. So if MAIA-10 is not transporting integrins to the Filopodia, then how do active integrins still preferentially accumulate only at the tip? At least they seem to be differentially regulated from focal adhesions. So if we use 2 of the widely used tricks in the field, either a CDK inhibitor or blebistatin that gets rid of mature focal adhesions, we still see active integrin localizing to the Filopodia tip, and this is the quantification you can see here. So clearly the Filopodia tip active integrin was independently regulated of focal adhesions. So then to address this, Mitra Mihkinen, a PhD student in the lab, went and took all the integrin activity regulating proteins that we found from the Filopodia tips and have been published in the literature, and also the inactivating proteins, and silenced them one by one to see which ones are essential for regulation of the integrin activity. And much to our disappointment, this didn't turn out to be super exciting, because the only other thing in addition to myosin 10 that significantly reduced integrin activity at the tip was double silencing of tailin 1 and tailin 2. Interestingly, for instance, silencing of kindlin, even both of them didn't do anything significant to the integrin activity, even though kindlin is highly localized to Filopodia tips. So how does then this co-function of myosin 10 and tailin function to regulate integrin activity in the tips? So one of the experiments Mitra did was that he started deleting bits of myosin 10 that are known to be involved in integrin binding. And what we was able to show is that if you delete the firm domain or the fragment of firm domain that's known to bind to integrin, you lose the integrin activity at the tip. So integrins are still localized to the Filopodia, the inactive ones, but you don't have integrin activity anymore. So clearly the firm domain was somehow needed for activation. Interestingly, and I'm not showing you the data, myosin 10 actually inhibits integrins. So when myosin 10 firm binds to integrins, it doesn't activate, it actually inactivates integrins. So that wasn't the mechanism. What happens if we now switch the firm domains between myosin 10 and tailin? So Mitro made this chimera construct, and together with Guillaume they imaged it, and what we could see was that in fact the hybrid myo-10 tailin forms Filopodia just as well as the wild type. The inactive integrin was still localized throughout the shaft, and the active integrin was localized to the tip. The tailin was a little bit more efficient, the tailin firm myo-10, than the wild type, but in both cases we had integrin activity at the tip. So I don't have time to show you all of the data, but the conclusion from our study was that in Filopodia tips, integrin activity is slightly differently regulated than other adhesions. And there's a two-step process in getting integrins activated at Filopodia tips. So what we think is happening is that we need a firm domain coupled to a motor that tethers the integrins at the Filopodia tips for sufficiently long to then hand this over to tailin and kindlin at the tip of the Filopodia to have the integrins activated there. It doesn't have to be myosin 10 firm domain that's coupled to the motor, because if we put tailin firm domain on the motor, it works equally well. So you first need to tether the integrin, and then you need to hand it over to tailin to get the localized activity. But the myo-10 is not needed for actually transporting the receptor there. So then I'll switch gears from Filopodia and regulation of integrin activity to discussing how force and stiffness is linked to regulation of cell migration. So as many of you are probably aware of, matrix rigidity regulates integrin-dependent adhesions, and integrins are important sensors of matrix rigidity. So if a cell is on a soft environment, the forces that the integrin and the tailin and vinculin are experiencing are so low that tailin isn't fully extended, and vinculin, which is here in orange, is not properly recruited to enforce the actin binding to the integrin. In contrast, on high force and high rigidity, the forces build up on the integrin bonds sufficiently highly that the integrins cluster, tailin is extended, vinculin is recruited, and these adhesions are reinforced. And this obviously has important implications for things like gap nuclear localization, proliferation, and many things that are related to rigidity and cell behavior. And this is also one of the reasons why, alongside the well-established hallmarks of cancer, it's also now widely appreciated that different physical properties are also important hallmarks and are also contributing to malignancy in cancer progression. So we all know that cells migrate and they respond to different attractive cues that are present in the tumor stroma, for instance. So stiffness can recruit cell migration, and this is a process called Durotaxis, where cells migrate towards increased stiffness. Jim Baer last week did beautifully what haptotaxis is, and then obviously with soluble ligands, chemotaxis can also attract cell migration. In this beautiful paper from Ken Yamada's lab a few years ago, they looked at a high number of the most widely used cancer cell lines. And what they observed was, first of all, that Durotaxis only happened at fairly low rigidity. So if they went above 18 kilopascals, they saw that there was much less Durotaxis anymore. The response to increased stiffness was lower. But at this sort of rigidity, what they saw was that all of the cell types preferentially migrated towards stiff versus soft. And we've been interested, obviously, on stiffness for a number of reasons. And we wanted to generate this system where we could have like a poor man's system to analyze responses to different stiffnesses. I won't have time to explain the whole process. It was published last year in JC, Journal of Cell Science. But what we basically do is we mix a soft acrylamide hydrogel and a stiff acrylamide hydrogel next to each other on a spot. We overlay them with a glass and we generate a gradient. And the way how we see where we are on the gradient is that the stiff gel contains fluorescent beads. And then what we've done in this paper is that we've actually come up with a calibration curve where you can just analyze the number of beads, and from there you can calculate back what stiffness gradient which part of the gradient you're on and what the stiffness approximately is. However, what we wanted to do with these gradients, in addition to analyzing other cell responses to stiffness, was to ask the question of, do all cells actually undergo durotaxis, and is the behavior the same across cell lines? And Aleksi Isomursu, a talented PhD student in the lab, was playing around with these gels, and he was plating different cancer cell lines on them. One of the cell lines we use a lot, and many of you and I know also use, is MDA-MB231 cells, which is a highly migratory triple negative cell line. And as you can see, they nicely make enforced paxillin and vinculin positive adhesions on the gels. And when we plate them uniformly on the gels and we'll allow them to migrate, we can see that they preferentially accumulate to the base, to the stiff areas of the gel, and this is the quantification here. However, when Aleksi then took another cell line and looked at its behavior, he noticed something unexpected. So a glioma cell line called U251 actually preferred to accumulate in the sort of intermediate stiffness areas on the gels and not on the 21 kilopascal stiffest areas. And that's the quantification here. And this is the comparison with the 231 cells. So you can appreciate that they actually behave rather differently. He then went to David Odie's lab to study this in more detail and also to undertake some modeling with Dave Odie's team to understand what's going on. And they had this slightly more sophisticated system where you could use UV lithography to generate clearly defined soft and stiff areas. And so they generated gels where you had fibronectin lines that traversed areas that were either 15 kilopascals or 8 kilopascals. What you can appreciate here from the video, hopefully, is that the 251 cells don't really like the 15 kilopascal stiffness. They go to the border and they turn back. And this is the quantification. So at the end, you start to see that the cells are actually centering themselves towards the soft area. And this is, again, the quantification. So what's going on here? So many of the cell types that people have studied in the field, and here's a list of some of them, they actually show this kind of uniform response to stiffness. So when substrate stiffness increases, the traction that is exerted by the cells to the substrate also increases in a linear fashion, or almost linear fashion. And this has been linked by studies from Pere Roca's group, but also others, to the ability to actually reinforce and make these stable adhesions. Now, what Aleksi observed is that, funnily enough, the 251 cells actually don't show adhesion reinforcement. So if you look across different stiffnesses all the way to 60 kilopascals, we don't see clear paxillin positive focal adhesions forming. If he also looks at the other mechanoresponsive phenotypes that people look at in cells, for instance, nuclear YAP, you can see that the 231 cells, they had this adhesion reinforcement, and they also have increasingly nuclear YAP with increased stiffness. And in contrast, the 251 cells are poor or have no YAP activation in response to stiffness. So somehow these cells that were not durotaxing towards high rigidities are also responding to rigidities with their adhesions in a different way. traction force microscopy comparing these two also revealed a big difference. So the 231 cells behaved like I described that many other cell types do. The forces increase with the increasing substrate stiffness. In contrast, the 251 cells actually have a traction optimum around 8 kilopascals, and then at higher rigidities, the tractions actually drop. And interestingly, this traction optimum maps very nicely with the area where we saw that the cells preferred to migrate to. And so the OD lab is very well known for their work on the molecular clutch model and modeling of this. And what they published a few years ago is a sort of upgrade of the clutch called the cell migration simulator, where they model which one of these protrusions wins based on the parameters that the computer is given. And this then defines which direction the cells migrates into. And so Jay and Dave and Alexi took upon themselves to then use the cell migration simulator to try to understand what's going on with the 251 cells. And what they could see in their stimulations is that if initially they put half of the cells on soft and half of the cells on stiff, and they give the cells the same parameters we had experimentally defined for the 251 cells, you can see that the simulation after 16 hours shows that the cells actually prefer to accumulate to the stiff areas, the soft areas. And this is the quantification. And so the result indicated that the cells actually prefer to migrate to an area that corresponds to their stiffness traction optimum. We had a careful look at the 251 cells, and they still have vinculin, they still have tailin. That's not the problem, and we don't fully understand why they don't reinforce their adhesions. But then we made an attempt to try to make the 231 cells behave differently. And one of the key factors that regulates this uniform traction in response to stiffness is tailin. And work from Zavi Trebat and Pere Roca have shown that if you deplete tailin from the cells, they actually start to show this biphasic response to stiffness. And so what Alexi did was he partially silenced tailin 1 and 2, because if you get rid of them fully, then you have an issue with cell adhesion. But with this partial silencing, he could nicely see that the cells are still spread, but they now have only very small adhesions, and the adhesion reinforcement is impaired. And this is the quantification. And so when Alexi now plated these cells on the substrate gradients, what he saw was that instead of migrating now preferentially to the stiff areas, they were actually accumulating in the intermediate stiffness areas. And this is the careful quantification of many gels here. So partial silencing and loss of adhesion reinforcement had now switched the Durotaxis response in these 231 cells. And so the model we have at the moment is that if you have adhesion reinforcement, cells prefer to migrate towards stiff because they can make these stable, strong adhesions. However, if you don't have this, you start to get frictional slippage on high stiffness, and the intermediate stiffness is actually the area where you have the right balance between load and fail. And this results then cells preferring to migrate towards their traction optimum. And so what we think we found is that actually some cell types are capable of reverse Durotaxis or negative Durotaxis, and they can migrate preferentially towards softer environments. We can explain the negative Durotaxis by motor clutch dynamics. So cells tend to migrate towards their traction optimum. And there might be a similar mechanism in other cell types, but this is normally overshadowed by the tailin vinculin mediated clutch reinforcement. And so with that, I'll just thank the amazing people that have contributed to this. So all the Shank work that was done by Johanna. This is from our lab retreat in the autumn to Lapland, where Alexi's dad, who's a keen bird ringer, helped us ring some birds, and this was really quite amazing. So Alexi was the main driver of the Durotaxis work, and Mitro and Guillaume is now an independent PI. Mitro is about to graduate from my lab, were involved in the Filopodia work. And this is my lab. So in Finland, we've got actually 4 different seasons. This is in the summer from a PhD party. And this is about 2 months ago when we went ice skating, and it was actually pretty cold. And for the collaborators, the OD lab and people from University of Minnesota for the Durotaxis work, Guillaume for the Filopodia, and Ben Gold for the Filopodia work, Eija for the electron microscopy. These amazing past lab members who've contributed to some of the earlier work I described, my team members and my funding bodies. And thank you for your attention.
Thank you, Johanna. That was a really nice talk, covering a lot of very interesting things about integrins. Just for the audience, if you have any questions, you can either use the raise hand button to ask you to unmute, or you can type your question in the chat. Claire, do you want to?
Yeah. Hi, Johanna. Nice to see you.
Nice to see you.
Thank you for the beautiful talk.
Thanks.
So if you're saying vinculin talin, talin vinculin is the reinforcer, that means that if you deplete vinculin, you should shift the optimum. Is that what you find?
So we haven't tried depleting vinculin. What we have tried is we've tried expressing the VD1 fragment of vinculin in the U251s. And this is very preliminary. It's something we're now working on, but it does seem that we get bigger adhesions, and we might, hopefully with that, be able to make the Durotaxing cells Duratax, but that's ongoing.
So that's one way, and then you could go the other way. Yeah, making the other guys by depleting it. Yeah.
Okay, great. Thanks, Claire. From Bob Fisher, he asks, does the OD cell simulation model include a lack of adhesion reinforcement? Sorry, I just lost it. A lack of adhesion reinforcement / vinculin recruitment, et cetera, at softer substrates like the cells show, or is it more like a cumulative spring model?
Oh my goodness, you would maybe have to ask David Odie that. I've understood that it's the basic molecular clutch model where they have a number of motors and a number of clutches and all these different parameters. And then the migration simulator just had several of these, and then it measures who wins. But the paper is on bioRxiv, so you can maybe check those data there. I'm sorry, I'm the cell biologist here.
Thanks. Maybe I can jump in with a question. So I know the idea of that cancer metastasizing cells could be metastasizing away from primary tumor sites through Durotaxis, but one of the problems was always that the tumor itself is stiffer becomes stiffer as it progresses. But now with this idea of reverse Durotaxis, do you think this could be the one mechanism by which cancer cells metastasize away? So they go by reverse Durotaxis, and maybe you would need some mutation in vinculin or talin in order to make that happen.
Yeah, so I kind of had slides on that, and then I removed them because I thought I would take too long, but I was actually faster. So maybe I'll share this again. So I had this slide, why might this be important? And it was exactly what you were referring to. So first of all, there was this Durotaxis paper recently published from the Engler lab. And there, I think they're finding similar findings than we are. And one of the questions, I guess, is exactly this. So if you look at stiffness of breast cancer tumors, and this is obviously not our work, and you measure in different areas, then in the middle, it's actually very, very soft. You go towards more the periphery, it gets stiffer, and then the edge where you have the collagen deposition is very stiff. And then if you think about this idea that you have a cancer cell that's surrounded by this very stiff ring of, let's say, collagen, and then it needs to sort of migrate out of there into the stroma and maybe into the blood vasculature, it would have to be able to go through areas of different rigidities and not only directionally towards stiff. And this is why I think something that would be really interested for the field to ask in more detail is what happens when a cell is migrating over different matrices, first of all. So it needs to switch from, let's say, a collagen rich to a fibronectin rich to a basement membrane type of matrix. And when this happens, and then at the same time, the stiffness changes, how does the cell coordinate all this and figure out what it actually needs to do? And I think we should all kind of try to challenge ourselves a little bit by building models where we could manipulate ligand changing and stiffness and try to ask a little bit more complicated questions than just migrating on fibronectin coated plastic, which I guess we've all been a little bit guilty of. So yeah, thanks for the question, Anna.
Thanks.
Okay, great. Thanks a lot for that. Our next question's from Raphael Petrosian, says, thanks for an interesting talk. Just wondering, could it be that for different types of cells, different substrate stiffness ranges should be used for understanding their durotactic behavior? For example, could it be necessary to use a wider range of substrate stiffness for the MDA-MB231s compared to the U251s?
Yeah, definitely. And something we've also recently observed and had a small paper in iScience last year is that if you take primary fibroblasts from the mouse mammary gland, and these have never seen plastic, and you put them on something we generally think is very soft, they have full stress fibers, fully mature enforced focal adhesions at stiffnesses of 1 kilopascal or even less. And this could be related to the fact that they actually come from an inherently very soft tissue. And to me, it would make sense that every cell actually has a stiffness optimum. And that might be depending on where it actually came from. And we may have lost a lot of this information by now looking at cells that have been on plastic for 40 years.
Thanks.
That is a really fantastically good point. Is that published, Johanna, or is that just an observation you guys have?
That was published last year in an iScience paper where we looked at how sharpin regulates traction forces. And there we see that these mammary gland fibroblasts think soft is stiff. Yeah.
Fantastic. Thank you.
Welcome.
A question from Margaret Titus. Terrific work, and she wonders whether if you have more information about the handoff to integrin between myotin and talin, how it could happen and what restricts this to the filopodial tip.
So we don't know how talin gets to the filopodia tip. Myotin obviously walks there alone on its motor, but talin also gets very highly enriched there, and we don't know why it's enriched there, but both of these are there. What I didn't have time to show you is that if we point mutate the integrin tail binding site on myotin, it still doesn't activate integrin. So it's really the binding of the myosin 10 firm domain to the integrin that's needed for the activation. We did try to do some competition studies between talin firm and myotin firm, and they were not fully conclusive. What we also find is that the myosin 10 firm binds both to the alpha and the beta subunit. And this is maybe one of the ways how it keeps the integrin inactive. And how it then gives it over to talin is still something we don't fully understand. It's not terribly easy to study, given that the filopodia, for instance, because they're so dynamic and so small, FRAP experiments and things like this are very, very challenging, if not impossible. But yeah, so we don't fully understand how the handoff works. Yeah.
Thank you. From Daniel Hammer, it says, dendritic cells use Filopodia for migration also. Would you know if anyone has studied which molecules are at the tips or shafts of Filopodia in dendritic cells?
Unfortunately, no. I don't know whether anybody's looked at that. Sorry.
Anantha asks, are there negative regulators of integrin activation on the filopodial shaft?
Yeah, so we were kind of hoping that when we silenced some of the known integrin inactivators, we would also see strong differences in the Filopodia, and that unfortunately wasn't the case. ICAP, which is published to be a beta-1 integrin inhibitor, localizes very strongly to the tip, actually. Sharpin doesn't really go to the Filopodia, except it's kind of there in the shaft. So what we think is actually myosin 10 itself is maintaining the integrins inactive, at least to some extent, because myosin binding to the integrin couples the alpha and the beta tails together and keeps the receptor inactive. But it's not localized to the shaft. So what's maintaining it inactive in the shaft and actually throughout the most of the plasma membrane is a good question.
Okay, great, thank you. From Durjay asks, is there any data available for such migration mechanism in endothelial cells like HUVECs in contrast to the MDA MB231s?
Related to the Durotaxis or the Filopodia?
I'm not sure, but let's assume yes.
So far for the negative Durotaxis, the 251 cells are the only ones that we've seen this inherently, and then in the tailin depleted 231 cells. We haven't looked at Durotaxis in endothelial cells. Endothelial cells do send, yeah, they do have Filopodia, but they're mainly in the junctions. Any cell type where you overexpress myosin 10 will make nice Filopodia. But endogenous Filopodia in HUVEC, I don't think are very prominent, and I don't think they express very high levels of myosin 10. So something we've known for quite a long time is that mutant P53 upregulates myosin 10 gene expression. And so this could be one of the mechanisms how mutant P53 drives metastasis in many different cell types. Including the 231 cells that actually have high levels of endogenous myosin 10.
And thanks. As a follow-up to that, Bob Fisher pointed out in the chat that endothelial cells in vivo have Filopodia and use CDC42, et cetera. So
okay,
just
yeah,
to follow
that's
up
a
on
good
that.
point. Yeah.
Thanks.
And just the last question from me before we wrap up. I was wondering how the integrin activation idea for in Durotaxis and reverse Durotaxis fits with, I guess it fits very well with Xavi Tripat's model of Durotaxis, which is about physical deformation of a substrate in order to decide which way you're going to go. I guess in this case, it's just you're physically deforming in the opposite direction because of a lack of reinforcement at the stiffer end. Would that be correct?
Yeah, so what we think is that the cells are slipping on when it's too stiff. And then when it's too soft, they're obviously not able to make high traction. And then for them, the optimal traction happens at 8 kilopascals, which then for some other cell type like the 231 cells, the optimal traction is higher with higher stiffness. Obviously, with the restrictions we have with the traction force methodology, I don't think anybody knows how high the traction actually goes. So I think our current idea that we have this sort of uniform buildup of force with stiffness is just because we can't go stiff enough to actually see the drop because of the fact that the traction force microscopy setups and the bead deformations and things like this just doesn't allow us to do that. But that's just my speculation.
Although I guess the stiffness range you're using covers a lot of the actual biological tissues in the first place, so there's not many that are stiffer.
Yeah. But then how biological these cell lines are because they've been on plastic for 40 years is a good question. I don't know, I've been talking to some people about whether you can educate cells to like soft or stiff. And I think these are really interesting ideas, but let's see.
That's what I was just going to ask. I wonder if it's possible to retrain them, right? Get them used to soft substrates by culturing them on soft for a few passages and see if they start to behave differently.
Yeah,
That would be a lot of fun.
I think we may have some observations like that, and I've heard other people have. But what I don't fully understand there, and I don't think we know before we do some proper single cell sequencing or something, is that is it actually that you have a heterogeneous population to start with? And there are some individuals already there that prefer soft. And then when you educate them, you're just selecting. Or can you actually change a cell so that you're not selecting for a rare subpopulation, but you're actually training the cells to prefer something? And this, I don't quite know what we're seeing and what other people are seeing, but definitely you can change behavior, that's for sure.
That's fascinating. Yeah. And not to mention that we've already been selecting for cells that are happy on the super stiff materials for years, right? So, all right, great. Thank you so much. Oh, and Bob Fisher also comments from the chat that it would be interesting to monitor vinculin expression as that education or selection process happens.
Yep.
Yeah, very fun.
351 cells have really a lot of vinculin. There must be something wrong with their vinculin. I mean, wrong in the sense that why isn't it reinforcing, because there's so much of it. It's actually higher than the 231 cells. So that's not the explanation. Somehow it's not, I don't know if it's not opening up or it's not getting recruited. So yeah, we don't know that yet.
Right, fantastic. Okay, I think that is all the questions that we have, unless anybody wants to pop in for the last minute. So I just want to say thank you again, Johanna. That was really fantastic. I personally have a lot of reading to go do to catch up on some of this. So that was great. And thanks, everybody. We will see all of you next week.
Yeah, thanks. Great having such a lively audience.
Thank you, Johanna. That was a really nice talk.